and we're so excited to welcome everybody here for our May Plants, Pests, and Pathogens. We have a really interesting presentation today about circadian rhythms and what those are in plants and how warming night temperatures are impacting that. Then we'll have our standard Be on the Lookout with Matt Bertone and Mike Munster of the Plant Disease and Insect Clinic here at NC State. So they'll be telling us about pest and plant disease issues to be on the lookout for in our gardens and landscapes in the coming months. And then Caroline will finish us out with our resource spotlight and Extension Master Gardener program announcements. So be sure to hang around for those. But I get the great honor of introducing our guest speaker today. And I'm really excited. I've been chatting with Colleen Doherty over the last year or two. We've been collaborating on some potential grant ideas or some ways that Master Gardener volunteers could be involved in projects related to her work. So that's still a work in progress. We hope to one day be able to announce something and invite you to participate in something. But today she's here to tell us about her work. And Colleen is an associate professor in the Department of Molecular and Structural Biochemistry, easy enough to say. She has a PhD in biochemistry from Michigan State University and completed a postdoc at the University of California in San Diego. Her lab here at NC State focuses on two main challenge areas. First, how can plants be used as tools to combat climate change? And then how are changes in weather patterns due to climate change affecting plants? That's what we're going to learn about today. She's going to talk about how these warming night temperatures, particularly, which is one aspect of climate change, are affecting plants' circadian rhythms and how that is causing stress on plants. So with that, Colleen, I'm going to hand it over to you. I will stop sharing so you can share your screen. And we are so glad to have you here today and eager to learn about circadian rhythms and what's going on with plants.
Okay, can y'all hear me?
Yes.
All right. Thank you so much for this invite. And as Charlotte said, I am very interested in collaborating with the Master Gardeners, and we've been putting in a number of proposals to try and make that happen on a number of projects. I actually don't have the little mini showing what it's showing, so or I didn't follow the instructions for how to get that. Can you see the presentation screen?
Yes, looks great. Thank you.
So I am really excited to talk to you all today, and I would really welcome if you have questions interrupting in the middle, because I would rather answer questions. I'll be happy to answer them at the end, but I'd rather answer them while I'm going, if that's the if you have a question. And please just shout out, since I may not see the chat. Okay. So my lab is, this will be the presentation overview, and I'm going to start with an introduction about recurring cycles and how those affect decision making. And we were just talking about lightning bugs and hummingbirds and some of these recurring cycles that happen, and that is very relevant to the core of my talk. So my lab is actually really interested in how organisms make decisions. That's our fundamental question, is how do organisms collect and process information to make decisions? So if you're faced with a tough decision, you want to get food, you want to decide if you need to move or stay where you are to get that food, you have to think about the environment, you have to think about the dangers and the benefits. And so I hope that it's possible for y'all to do this like interactive little things. So you can go to menti.com if you're on a computer, or if you're on your phone, you can take a snapshot of the QR code. If you go to menti.com, just enter this little code here. I'm going to switch to the next slide, but both these things will stay up so you can see them still. And this is kind of a question about how we process information and make decisions. And so I'm going to show you 4 pictures. And so as I show you the pictures, just enter, 2 words about what you see in that picture, because you're processing the information of the picture coming in, and then do that for each of the 4 pictures. Okay. Can somebody give me a thumbs up if you got the thing working? Hopefully we'll.
I have the thing obscuring the screens.
Oh, yeah, I'm sorry that's gonna be it. Yeah, I was
You should
having
be
that
able to,
problem.
you should all be able to drag it out of the way. So hold down and drag that window out of the way, and you could see both Colleen's screen and your whole.
I'll go ahead and go to the next slide. So you may not need the whole screen, so you can see just the first picture. Oops.
I put the link in the chat as well, if that helps
Oh,
anybody,
thank
so
you.
you can
That's
just,
great.
yeah, click
thank you.
directly there.
Okay, so some people said they got it. Okay, so I'm just going to spend like a couple seconds, just type in a few words. I'm just going to show you these pictures. So there's one. Here's the next one. And then the next one. And then the last one. And I know I went kind of fast there. You can still continue to type in your stuff, or if you remember those pictures. And take a second to write your answers. And while you're doing that, I'm going to work on being sure I'm going to have to end it and restart the show here to go to this. So we can see the answers that people have. And so what people have put in, the first one was probably the first one you had time to type stuff in for. So gorilla, rooster, buffalo, children, chicken. This is our typical response as humans. We see the danger and we see the thing that might be the most possible chance of threat to us. And so in all those pictures, the most possible chance of threat was, obviously the animal in the center. And if you think children aren't threatening, I am looking for a babysitter. So, you can come in and test that theory out. So, but what is really the major picture, the major pixel area of that was all plants. So if I go back through those pictures, this we're very interested in is how bias affects our perception. So here, most of the background here is plants. The rooster is the very small part. Most of the background is plants, but the bison is a small part. There's actually a fire going on there. Again, the plants are the major pixel area in these pictures. And yet we as humans very typically see the thing that's going to cause us the most danger, and we focus on that. And so that's, we call it plant blindness, but it's just how our perception works. It's just how we see things. Okay, so I will say that there were some comments about greenness in there, and I never get that comment. So I think that's because I'm talking to master gardeners who actually are catching some of that stuff. So that's cool. It's the first time I've ever seen the word green show up in there. So when an organism wants to make a decision, it collects and processes information, and that processing circuitry is biased based on what it thinks is going to be a threat. And plants are no different. Plants are very perceptive to their environment, extremely so. And so they're very perceptive to things that will be a threat to them, not just biotic threats, which we tend to be more perceptive to, but also abiotic threats. So drought, temperature changes, etc. So we're interested in how do plants collect this information and make these decisions. And you may think, well, plants aren't making decisions, but plants are continually making critical decisions. Most important they're making is whether to grow or not to grow, how to allocate that energy, whether they respond to a stress or they put that energy into going ahead and making seedlings, whether they're going to reproduce or try to stay vegetative a little longer and not reproduce. So as they make these critical decisions, one of the great things about studying plants is that they don't have a central nervous system. They don't have as much peer pressure to influence their decisions. And so really everything is hard-coded in the DNA. So if a plant makes a specific decision, we can actually understand that a little bit easier in the pathways that regulate that decision. And so that's really the question that drives my lab, but it's a little bit more complicated. So if we just simply ask, how do plants make decisions? And we're interested in temperature stress. If the plant is faced with some environmental change, such as an increase in temperature, does it make a decision to just continue growing and try and mitigate that stress, put energy into fighting that stress, so continue growing? Or does it change how it allocates its resources and just kind of give up and go to making seeds so that the next generation has a better chance. And this is something we typically see with plants. Once they go to make the transition to reproduction, they're not very stress responsive because they really have given up and putting all their energy, all their resources into producing the most successful seeds. So what we're really interested in is how do plants make good decisions in response to environmental changes? And we know that plants make good decisions because the plants and other organisms that make bad decisions don't remain in the gene pool very long. They tend to get kicked out, and they're not there to pass on their DNA with those correctly encoded decisions. And plants can make bad decisions too. And if they make a bad decision, then the chance of them passing on the DNA that made that bad decision is less likely to be either selected for by the gardener or selected for by nature. So generally, more information is thought to give better decision-making success. So this is a common theme in our decision-making processes. Collect all the information you need, write down a list of pros and cons. Plants do something very similar. They collect as much information as possible. But the more information you collect, you can start to get contradictory information. So you can get, for a plant, you can get sunlight telling you to grow, but a pathogen telling you you need to, you've got pathogen, you don't want to grow because you don't want to allocate those resources to that. At the same time, you might be having challenges with your nutrient availability in the soil and the temperatures dropping. So if those are telling you to do different things, how do you make a decision and integrate all of those signals and especially those that are conflicting? So now, to summarize kind of the overview of my lab a little bit more complexly, we're interested in how do plants process and integrate these multiple, often conflicting signals to make these evolutionarily advantageous decisions. So if I'd started off with that question, you would have been like, what? But really, it's very simple. Just how do plants take in all the information they have, evaluate them even when they're contradictory, and still make the right decision to be the most successful for their offspring and the next generation of seeds. And so just like us, if you're trying to organize and you want to be a very good decision making, time is a critical component of that. And so time management is what we really look at in my lab, and how time plays a factor in these plant decision making. So you've probably heard nothing can be certain except for death and taxes, but there's a few other things we can absolutely be certain of. One is that every morning the sun will rise in the east. And another is that approximately every 365 days, the earth will complete one complete revolution around the sun. And if those two things, either of those two things don't happen, you really don't have to worry about your taxes because things will be very bad. Because of these two recurring cycles, our earth is full of cycles. We're layers and layers and layers upon cycles. So we have cycles of biotic living things. So pests come in cycles. There's talking earlier about Charlotte was saying, though, the weather was just the perfect time for the pests to come, and now the plants have grown out of it. And so once you get past a certain stage, you might not have to worry about specific pests. Same thing with pollinators. The hummingbirds are out now. If the plants aren't ready, they'll miss that one chance for the hummingbirds to be here. And if you have allergies like me, the pollen, you have certain times a year when you're really struggling and other times when you're not. So those are biotic cycles. Those are just a few. There are many, many more. But we also have abiotic cycles, such as temperature, UV, precipitation, and this can happen in a daily 24-hour cycle, but also in a yearly cycle. So sometimes a day are more prone to be wet and humid than other times. Sometimes of year you'll get more rainfall, and these cycles are very recurring. And plants aren't stupid. They take advantage of these cycles. They know that these happen, and then they use those to their advantage when they're making a decision-making process. So if they get one of these complex conflicting signals, they can say, well, is it the time of year when the pathogens are going to be getting worse? Maybe I really need to invest my energy into defending against those. Or is it a time of year when the pathogens are almost gone? Maybe I can go ahead and just deal with these other stresses instead. So how do plants, know what time of year it is, know what time of day it is, anticipate that these recurring patterns are going to happen? This is all facilitated by their internal circadian clock. So just like us, plants have a circadian clock. Almost all living organisms on Earth do, not all, but almost all. The clock takes inputs. We call these zeitgebers. That just means time givers. And these inputs can be like the light coming on. For humans, one of our inputs is definitely bright light, but is also food. So if you're jet lagged, eating at the right time of where you want to adapt to is really important. So these inputs feed into this molecular oscillator. So this is the internal clock, and they control the biochemistry of the plant. They control the activities of the plant so that the right activities happen in time with the external environment. So the most common example is that plants photosynthesize during the day. Before the very first rays of light hit them, they have all their photosynthetic apparatus up and ready to catch the first rays of light. And they do that because the ones that don't, the ones that sleep in, miss that cue and get outcompeted by their neighbors that can outgrow them. So they anticipate dawn coming up, they're prepared, they have their photosynthetic machinery there, and then they shut it off at night because that's a very expensive use of energy. And if they don't do that, their neighbors that do shut their photosynthesis off at night have a better use of their energy and outcompete them. So this proper timing and ensuring everything is in time is well reinforced by evolution or selection in the garden. So the circadian clock controls, I want to say, all activities in plant. I have yet to find an activity that it doesn't control. When we write papers, we typically say most, just in case there's one out there we don't know of. But if you're looking at any particular activity, there's a good chance it's controlled by the circadian clock. So when the plants take up water, when the flowers open, what time of year they flower is controlled by the circadian clock. When they do photosynthesis, when they open their stomata and are therefore more sensitive to pathogens that will infect them through the stomata. Their response to biotic stress is very well regulated by the clock, as well as abiotic stress. So if we want to understand yield, we want to have beautiful plants, we need to understand the circadian clock. And so the clock does this by controlling the basic biochemistry. So DNA is what can be possibly encoded for. The plant cells have all of the possible responses that they can give, but the RNA is what the plant wants to do. So this is like what it's trying to do. These are the proteins it's hoping to make. And we can measure RNA really easy in the lab. So this graph here is from rice plants that are grown in the field. This is every transcript of RNA that's made. So each row is a transcript of RNA. And so this row here, this whole group of genes here, each column is a time of day that we took these samples. So we took these samples in the morning in this column, these samples 3.5 hours after the morning, these 3.5 hours later, these ones would be about dusk. So this group of genes up here at the top, these all coordinately peak right at dawn, and then they're off for most of the day. And then you can see them all coming up again right before dawn. They're starting, red means they're getting higher, blue means they're getting lower. So they're starting to come up again right before the next dawn. These actually happen to be all the photosynthetic genes. They're coordinatedly expressed to be coming up right before dawn, very highly expressed in the morning. By then the proteins are getting made, so the RNA doesn't need to be there anymore. And so they start coming back down. And so if you look at any column here, you can see that there's a different set of RNAs, a different transcriptional landscape for this plant, depending on what time of day you do your experiment, what time of day you sample the plants, what time of day you look at. So at this time of day, the plant has all these genes on and all of these genes are off. At this time of day, these 2 times a day, which would be like midday, all these genes are on, but now these morning ones are shutting off. So different times a day, completely different plant. Same DNA, but completely different responses going on. This isn't true just at the transcript level, it's also true at the protein level. And the really cool thing is that this manifests as physiological responses. So we can see here, these leaves are, opening and closing on a regular 24-hour period. And I love this picture of this pumpkin down here at the bottom. You can see at night, it really balloons up and then it stalls during the day and then it fills up again the next night. And you can see this rhythmic pattern of the pumpkin, filling up. And if you look, you can also see rhythmic patterns of the leaves, the circumnation. that's not quite the right word. I didn't say that quite right. And even the flower opening and closing and senescing has a rhythmic daily pattern to it. And so what's happening is the plant, through all these biochemical changes, is at night allocating the resources that it has left over. It did photosynthesis all during the day. It put all of its energy into that. And then at night, it says, well, how much do I have left over that I can feed into this sink, this pumpkin, and let it fill up? And so it's making those decisions at night about how to allocate its resources. And during the day, it's actually focusing on photosynthesis. So we call this kind of allocation circadian gating, and circadian gating can control how we respond to the environment. So back to that decision-making question, for us, for mammals, if you eat at a certain time when your liver is ready to process, you can eat a very high-fat food, you can eat a Western diet, lots of fat, lots of sugar, and if your liver is functioning and active, you can process that and not gain weight. However, if, like me, you tend to snack at night, and this is a very bad habit, and I'm in the circadian field, and I know how bad this habit is, you can eat the same high-fat Western diet, and your liver's not ready to process it, you don't have the correct processing, and you gain weight. And so this is called circadian gating, where the very same stress, the exact same environmental input, in the exact same genotype, the exact same person, the exact same mouse, can cause a very different response. So we're not changing the DNA here, we're not changing the species, we're not changing the input, we're just changing the time of day. And so this circadian gating is true for a lot of different components, many different environmental stresses. One of the most well-studied in both humans and plants is response to pathogens and pests. So just like for humans, and another one I like to throw in is our cognitive responses are very circadian gated. If I give you, a quick reaction test or a math test in the middle of the night, unless you're very unusual, you will perform significantly worse than if you do it in the late afternoon, mid-morning time. So all of our responses, even to stimuli such as math stress, are circadian gated. And so this is true for plants. UV, photosynthesis, I already said, plants won't photosynthesize at night, even if you give them a really bright light, because their circadian clock is telling them not to bother putting it up. They don't expect the light then. But also temperature, stress, and pathogens. So perhaps the best studied is pathogen stress response and the time of day and circadian gating of that. And so if we look, for example, at an herbivore, so a stress responsive herbivory, if you take, there's a classic experiment where they took these worms called teeny, and they put them on tomato plants or Arabidopsis plants, and the plants can fight them off. They're a generalist herbivore. They don't really hurt the plants. The plants can produce some stuff. The bugs will take a few bites, but they'll both kind of exist, but they won't overrun the plant. However, if you grow them in separate chambers so that the caterpillar thinks that it's day, when the plant thinks that it's night, and then you mix them, the plants will get decimated. And that's because the plants don't expect to see that caterpillar at night. And we don't actually understand why, is that the plants can't sense them, or their biochemistry system just isn't there to allow them to mount that response. But whatever the case, at one time of day, if the plant sees this caterpillar, it can mount a very robust response. It can keep the caterpillar from eating it to nothing. But at another time of day, it can't do that, and it just gets decimated. So this is like my favorite example of how the clock is very robust in controlling stress responses. In my lab, we're very interested in temperature stress. So we expect that days are almost always warmer than nights. There's about 3 times a year in Raleigh that flips and the nights are actually warmer, but it's not that common. And so we had this hypothesis when I start my lab that heat stress would be gated by the circadian clock or the time of day, so that the plant would be able to mount a very robust stress response at one time of day and kind of a muted stress response to temperature at another time of day. Excuse me. So I need to explain a little bit about the experiment we did. So if you take the plant we particularly worked with, you take it out of its happy controlled environment chamber where we grow it, and you suddenly give it a heat shock, the plant we work with can survive up to about 42 degrees. And if you go over 42 degrees, it kills over and dies. So that's like its basal temperature limit, just going from normal conditions to heat stress. That's like the break point for it is about 42 degrees. Plants also have acquired thermotolerance. So they basically, if you take your plant from a happy, cool spring day, and then you give it a mild but non-lethal temperature stress. So high about 38 degrees, a warm summer day, so 90 degrees, then you can actually increase its tolerance. So instead of dying at about 42 degrees, it gives about 5 more degrees Celsius increase in tolerance. So it's quite a significant increase. And we call this acquired sensitivity. So their ability, their acquired tolerance, how they can adapt with this mild temperature signal. So to me, this whole system is really cool. The plant senses, oh, it's getting warmer. It has to make changes to then allow it to withstand even higher temperatures. And so that process is really working. Okay. I'm sorry, something's popping up for Zoom. I don't know. Okay, so the experiment that we did was very simple. Just take the plants from their warm, happy chamber at dawn, midday, dusk, and midnight, and expose them, either take them straight out of their chamber and put them just to 22 degrees. That's our control. Just move them to room temperature, or move them to 40 degrees or 42 degrees or 44 degrees and see which temperature kills them. So we did that at these 4 times a day, and that would answer basal temperature tolerance. Does basal temperature tolerance vary based on the time of day you do your experiment? And then we also took some plants out of the chamber. We gave them 1 hour at this non-lethal temperature. We let them rest for an hour, and then we moved them to one of these to see if the time of day affected their acquired thermotolerance. So before I show you the results, which is the next slide, I predicted that the midday would be the most heat tolerant because those plants are, in the middle of the hot sun, they're going to be very ready, and midnight would be the least tolerant because it's the middle of the night. They're not going to, be expecting much heat. They'll drop their tolerance then because it won't be a good use of their energy. That is absolutely not what we saw. So this chart here, each of these is a Petri dish of plates. It's zoomed way, way out. So this is a bunch of plates. And these were the plants that were taken out at dawn, midday, dusk, and midnight. And then this was the plants that were moved to 42 degrees. You can see everybody managed to survive that. And then 44 degrees, 45 degrees, 46 degrees, 48 degrees. So everybody pretty much dies at 48 degrees. However, and so the minus are the ones that were taken straight out of their chamber and put right to the temperature. They didn't get any acclimation, so they're minus acclimation. And then the plus ones are the ones that got the acclimation. And I told you, if you get acclimation, you can actually increase your temperature tolerance. But here at dawn, they don't do that. We don't see that. You can see that at midday. You can really see it at dusk, and you can really see it at midnight. So my prediction that midday would be the best and midnight would be the worst was completely wrong. I made the students redo this experiment about 20 times because I didn't believe them. But many different students and many different postdocs have repeated this, and we consistently see that at dusk, the plants have a really amazing ability to increase their tolerance. And that is really bad at dawn. The plants are really insensitive. At midday and midnight, they're actually pretty equal. There's not much difference between those two. This is the same thing over many experiments. So this is just one experiment here. And these bar charts are just quantifying that over many experiments. Dawn is the worst, both in the basal, which is in yellow, and in the red, which is in the acclimated. And you can see at dusk, they really have this amazing response. And they're able to acclimate very well, and they have the highest tolerance. So this answered our question. Yes, temperature response is gated by the circadian clock at the physiological level. And so that was an important question to answer. The answer wasn't what I expected. And they're actually the most tolerant to high temperature at the end of the day. We've been thinking about that. We have some ideas why. But now I mentioned that, that pumpkin blowing up, that's all controlled by the underlying biochemistry. So we wanted to go back and ask, well, what's the underlying biochemistry going on? And so because we can measure transcripts, we just looked at that because that's the easiest. And because we didn't have a lot of money, we just did 2 times a day. We did the dawn, where it was the most sensitive, and we did the dusk where it was the most tolerant to that stress. And we just took them out and we just looked at that low non-lethal heat stress temperature because we wanted to see what's changing there between dawn and dusk that is allowing the plants to really give this robust stress response at dusk and not in dawn. And what we saw was that so this is an example of just 2 genes here. So this is the level of this gene in the morning. This is the level of this gene in the morning after heat stress. So you can see this gene goes up over 1,000-fold. This is a very responsive gene, but it's only responding in the morning. Here's the level in the control at night, and here's the level in the control at night under heat stress. It's responding at night, but it's puny compared to this massive response in the morning. And most of the heat response genes that we know about, and this is a well-studied field, match this pattern. They're very responsive in the morning, not so responsive at night, but yet the plant is way more tolerant at night. And so we do see genes that, maybe respond a little bit in the morning, but are very responsive at night. And we're very interested in following these up, particularly because these genes have not been associated with heat stress response before. And my opinion for the reason for that is that most faculty don't make their students come in the middle of the night to do their experiments. So we have this really unique perspective of what the plant is doing at night and why, what genes are changing. And these are really unstudied genes. Most of these genes are unknown. We don't know anything about them. And I think that's our bias as humans because we're diurnal creatures. So coming back to that idea of bias, we only see the tiger, the gorilla. We only see the things that are responding in the day because we have our own internal biases. So to summarize this section, and then I'll get to high nighttime temperature briefly, is that each temporal perspective is unique. So we looked at 2 times a day, but if we broke it up into finer points, each slice will give us a different response. And so if we want to understand plants, we can't just look at like the times when it's convenient for us, right after breakfast, after we stroll in the lab and have our coffee. We have to look at many times a day to understand the full response of the plant. And we think a lot of these night responsive genes are going to be unique and largely unknown. And comes to this idea that we really don't understand what plants are doing at night. One of the things I really want to interact with the master gardeners about is how much do you know about plants at night? And, how much do you know about your garden at night? And how are things maybe different between that? And so I think the bottom line is that our experiments and our understanding of plant stress response is biased by our own clock, which tells us we shouldn't be up in the middle of the night because our cognitive abilities are worse, because our sight is worse, because we have lower temperature and higher heart rates at night. So those are things that affect our ability to respond. So what does that have to do with climate change? So this is the last little bit here, and I won't go into super detail, but I mentioned that plants use these recurring events to properly time their stress responses. And it's not just the time of day, but also the photoperiod, so the season. They have these daily temperature cycles, the memory of the previous exposure. But one of the biggest challenges I see with climate change is that the timing of these recurring events is changing. So we're having warmer nights, earlier springs, off-season heat waves. Plants are very adapted to expect it to be hot at a certain time. And now if it's getting hot earlier, they may be flowering then, or they may be setting seed then, doing these very precious temperature sensitive activities that they've arranged to avoid that heat stress. And now the heat stress is coming earlier. And so, I'm probably preaching to the choir here. Y'all probably already know this better than me, but, hardiness zones are moving northward. And so the same plant will now be getting heat at a different time of day in its developmental season. So it's because its growing zones are moving northward. We also see pests and pathogens also moving poleward. So the organisms are moving poleward. So plants grown in North Carolina may be seeing new tropical pests that they've never encountered before, because now those pests can also have a hardiness zone that's higher. Same thing for invasive species. Also means we can grow plants here that we couldn't in the past. So there's some benefits as well. There are some benefits to this. Longer growing seasons is one of them. We also see earlier springs. So this is a chart of the annual start of spring, and the green indicates it's earlier than the average, and the blue indicates it's later than the average. And there's some noise here, but generally you see the green is really progressing here towards the end. And then my lab really focuses on this asymmetric warming of daily temperatures. So just the last point here is that, the plants have these biochemical pathways well encoded to properly time their events. It's really important for their success. But now those pathways aren't getting them to respond at the right time because these things are now not happening when the plants expect them to. So as an example of just kind of how the plant clock works, these are some traces of a gene expression. So this is, again, that RNA, because we can measure it very easily. And so this is the night, day. So this is dark, light, dark, light. And then if we move the plant into constant light, so this is subjective night. So the plant thinks it's night, but it's really still got the lights on. We can see that this pattern persists. So we can entrain the plant. It has this very nice 24-hour pattern, and that will persist. And the same is true for that like pumpkin blowing up. Even if you let it go into constant light, it will still allocate its sugars at the end of the night, because that is the circadian clock. It's not driven by the light cycle. The light cycles help to keep it on time, but it's really driven by the clock. So if we look at this in these circadian conditions, about 60% of the genome shows this, in the stale conditions, about 60% of the genome has this pattern, and about 30% continues to persist. So we say that, light cycles can entrain the circadian clock. But what's been recently discovered is that it's not just light, it's also other things. So for humans, it's not just light, it's also when you eat. That's a major discussion in the circadian field in humans right now. And for plants, it's temperature cycles. So we know that temperature cycles can also entrain the clock. And that's a problem because with climate change, nights are warming faster than days. So while both the daily maximum and daily minimum temperature are increasing, the minimum temperature is increasing faster, so the daily temperature range is decreasing. And what that means is that we have a reduction in yield, in grain quality, and that's really what my lab is working on and how that's happening. I'm wrapping up because I know I'm right at the end of time here. So, sorry, it's not going forward. I think the next one's a big picture, and that's why it's stalling on me. Sorry, oh, I went too far. Oh. So we did this experiment where we grew plants under, this is our rice plants, this was grown in the Philippines. These are plants, these are control plants in normal nighttime temperature. And then we have these plants in these ceramic rings of ceramic heaters, and they're heated only at night. So here's the day temperature, the control plants are in blue, the warm nighttime temperature plants are in red. And you can see there's no difference in the day temperature, but the night temperatures are getting warmer. And so we're never getting a heat shock. They're never getting a high temperature. They're just getting a little bit warmer at night. And with that, we see a substantial decrease in yield, as has been seen by other people. This is the grain yield, dry weight. Basically, it's really bad news with just a 2 to 3 degree increase in nighttime temperature. And just to give you one example of a gene that shows this effect, this is photosynthetic gene, as I mentioned before. The plants start anticipating dawn. They start turning these genes on, and then they come up during the day, and then they shut off in the afternoon, and they have this cycle. Under warm nighttime temperature, these ones are a little bit more like teenagers. They sleep in. They try to overcompensate. They're actually going up to a higher level, but then they kind of peter out again in the afternoon. So under warm nighttime temperature, that very tight coordinated cycling that I showed here before, where all the photosynthetic genes are coming up at the same time, is now distributed throughout the day. So that very tight pattern is really disrupted. And so it's not just true for the photosynthetic genes, but for almost all transcripts in the entire genome. And so these warmer nighttime temperatures are really bad. This very small increase that we have already seen in the last 20 years, and we are expected to see in the very near future, we're not talking about a 1 degree increase overall, but just at night, can have this substantial impact on rice yield. It's also been shown to work on having a negative effect in wheat and sorghum. And interestingly, warm nighttime temperatures also are beneficial to some pathogens. So we're interested in how these warm nighttime temperatures negatively impact the clock, but also positively impact other stresses, and so how that interaction works. So I would like to thank the students in my lab who are really good, Kanjana, Edmeritz, and Jigger. All my students are really good. I shouldn't say it like that, but these are the ones that had to come in the middle of the night and do all these experiments. And then I have collaborators in the Philippines and Kansas State and other collaborators here on a project that I didn't tell you about, but hope to collaborate with master gardeners about someday. So I think I can escape. And
Excellent. Thank you so much, Colleen. That was a fascinating presentation. And I think we'll have some questions coming into the chat. I know I personally just thought of 10 questions about how you did these experiments and all that cool stuff. So one of my questions, just to start off, was relating to the temperature that you stressed plants out to. It seemed like you're speaking in Celsius, and so it would seem like it was between like 107 and 117 degrees Fahrenheit. And maybe behind that, what was behind that particular temperature is are you thinking about that we've got radiant heat in addition to really what the weather forecast might say the temperature is, or just where you found that particular heat
why
stress
that
level?
temperature? Yeah, so
Yeah.
the question, why that seems like an extreme temperature we're never going to have, right? But most of my work is in crops, although I've my other project, which I didn't tell you all about, is in pokeweed, and so I'm interested in the other. But in the canopy, it does reach those temperatures. It's maybe not here in Raleigh, but in Kansas and a hot summer day, it probably does in North Carolina as well, because the plants give off additional heat. It gets very hot in that canopy, and it is dependent on the plant. Some plants are actually, rice is way more temperature tolerant than other monocots, other species. So it kind of depends on the plant and the temperature there. But we also use those because it's a little bit artificial, because we want to probe like what are those pathways and where are their breakpoints. But that 30 degrees where they're making that transition, that's what allows them to survive those like extended heat stress, because it's not just we're doing these. The other reason for those really high temperatures is we want to do them in short bursts, because we want to look at like how the time has an effect. And temperature has multiple components. It's the time when it happens, which is my interest, but it's also the intensity of the heat, the duration of the heat. And so we wanted to keep that duration small to focus on discrete time points to make the experiment easier to do. But the plant can also, the lower temperature over a longer time can have a similar effect. And we have done those experiments to demonstrate that it is the same, that they're more tolerant to a longer stress of lower temperature at night than they are in the morning.
Oh, that's so interesting. And one of your slides, I think, was talking about that stress point, and that a plant, if you sort of gradually take it out or take it to a higher temperature, it can acclimate easier. And I think that called up to us the idea of hardening off our seedlings in the spring, right? We bring them out gradually, we try not to stress them out, and then they can handle it. That's really
exactly
the same concept?
Yeah, it is exactly the same. And it's also true for light. So there's this acclimation response. My lab calls it memory because of the specific one we work on, but in other places it's hardening. It's this, this acclimation response that there's the plant is getting the signals, right? It's a decision pathway, which is so fascinating to me. The plant's getting a signal that says something's happening, I need to prepare, and it does it. Like in the case of hardening for light, it makes more chloroplasts so that it can handle the excess light that it's going to get and not have too much reactive oxygen. And there's a question about like the light staying on all the time in the sunroom. And actually, yes, constant light is not good for plants. They need a rest period. We don't call it sleep because we get in a lot of trouble with that, but they actually will do better if they get a slight break at night. I mean, it depends on, what your goal is, but if you want, from better, I mean, more greener leaves, more photosynthesis, more response, more tolerant to stresses, even a short break of like 2 hours is enough. So light at night. So when like people try to grow strawberries or things in greenhouses for yield, they really try to grow them like constant light. And it turns out that's worse than going down to like 4 hours off, and it saves electricity. So there's, And
Right.
probably the best study to this is the people who grow marijuana indoors, right? So they want the best deal, whatever. And it turns out that they figured out also that, a little bit of a night break is really good for the plants.
Yeah, that reminds me, Colleen, this is Charlotte. Was talking with a master gardener yesterday, and we were talking about outdoor lights, the outdoor, safety lights. Do they impact plants in that same way, like you were just talking?
Yeah, so light at night is a really interesting question because the plants, expect it to be dark. And so what does that signaling do? If you shine, moonlight on them, they won't start photosynthesis. If you shine a bright light on them, they won't start photosynthesis. But if you do it over and over again, that can reset their clock. And so we don't really know. These are all questions that haven't been answered. Like, so humans, we're in a state of called chronic jet lag because we don't get bright light during the day and dark light at night. We get this blue light, and there's tons of studies about circadian disruption in humans and all the things that causes and all the problems. But how about plants? Circadian disruption in plants, does this light at night affect them like it does us? And my hypothesis would be 100% it does. There's a couple of questions in chat here, but my glasses aren't very good. And
No worries, I'll help you. One of them is wondering about, would there be an ideal time of day to fertilize, given that you're talking about nutrient uptake?
yeah, so when to fertilize and when to water is really, people always say water at night because you don't want to burn off, right? That makes sense. But is that the best time for the plant? And that probably is the best time if you're just watering, an open field that can evaporate. But if you're watering something that won't evaporate, is that the best time? We don't know. And then the same thing for fertilizer. We know that it makes a difference. So one of the things my lab studies is when you spray, I can't remember the other name for it, Basta, Roundup, it actually won't work if you spray it at certain times of day. So the time of day you spray herbicides makes a big difference. And that depends on the herbicide, but it does make a big difference. And so fertilizer would be the same thing if it's there. So generally plants do most of their transpiration at night. So that's probably why watering at night is good, although we haven't actually tested that. So my guess would be fertilizing at the end of the day would be better because that's when they're more likely to take it up. But again, these haven't been tested at all.
Same thing with relating to taking cuttings for propagation. Someone wondering, when is that plant maybe at its best? So this is really fascinating.
So based on our work for the high temperature, I would say putting them outside at night, they'll be more able to survive it, because then they'll have that heat shock will be at night and not during the day. Now cold is completely different, right? So when you want to move them out and it's going to be cold, that would be a different.
Yeah, and I think this person might even be talking about when you're actually taking cuttings for propagation. So when is the plant the most turgid? When is the plant, the least stressed? Or
yeah. Sorry, I missed that. Yes,
also
possibly.
interesting.
Yeah. And the question about the hydrangea and light every night. So this I always get people arguing, I have my plant in constant light and it grows really big. Yes, absolutely. There's two components here. More light equals more photosynthesis. The plant will grow until you do it. But the question is, if you gave it 18 hours of light all year, or 24 hours of light, or 20 hours of light, I would predict, my hypothesis would be that, 18 hours, maybe 20 hours will be much better than 24 hours. And but 24 hours is probably a lot better than 8 because it's just getting a lot more light, in the early spring when the other plants aren't. So there's some, balance there between just total photosynthesis that's going to overcome other things and light at night. But yeah, circadian disruption of plants is a whole new field. It's also a whole new field in humans, but it's becoming, really scary how much we're disrupting our clocks through staying up late, eating late, not getting bright light during the day. So I think y'all are probably some of the healthiest people if you're out in the sun, getting really bright light. Just be sure the lights are off at night. Yeah, what time would it be best to transplant hardened seedlings? We haven't done these experiments. This is exactly why I want to work with the master gardeners, because we could ask this, we could transplant some of them, do enough at different times and in different locations and say, my hypothesis would be night would be the best, but nobody probably does that. And so it'd be interesting to see if that would.
Yeah.
true or
Well,
not.
you have piqued our curiosity on so many levels, and I think it's exciting that, in some ways there is so much research to be done. There's so much learning out there, and it's great to know your lab is doing this, and that you're going to need to torture some more students with nighttime lab work.
They know
Just
when
part
they
of the agreement.
when they join my lab, they know that's what we do. So yeah,
Excellent.
I let them have the
Well,
next
Colin-
couple of days off to recover.
Good. Yeah, you got to get their circadian clock back on
Yes.
track, right? Well, thank you so much, Colleen. That was a really wonderful presentation. We've learned a lot, and we've got even more appreciation for the complexities of plants and the complexities of the environments that we put them in as people, and then just in terms of how the globe is changing and the weather is changing. So we
Thank you
appreciate your
so
time.
much.
Lots of compliments coming
This
through.
was really fun. Thank you very much. This was a fun talk to prepare and fun to give. Then fun questions. Thank you.
All right, and hopefully we'll be back in touch maybe about some projects sometime.
Fingers crossed.
Yes, great. Thanks, Colleen. All right, Matt looks like he's getting ready to share his bolos. I'll turn it over to you, Matt.
Sure. Mike, you want me to go first?
Go for it.
Sure. Okay, to get this started. Okay. Well, thanks everyone for coming today. That was a great talk. I really love the integration of the research, and actually it was pretty easy to understand as far as, I think practical applications of that. And that actually affects our bolos because, these are very general kind of things we're saying, especially different parts of the state and different seasons will cause, or at least timings of seasons and warm temperatures, rain, things like that will cause different arthropods and diseases to be more prevalent than others, different years and in different regions. So these are very general bolos. Be on the lookout for June. But one is going to be lots of young caterpillars, including everyone's favorite, the bagworms. So I like to show this picture. This one little small section of arborvitae that was submitted had about 30 little tiny baby bagworms on it. So if you have plants that seem to be attacked by them, or you're worried about those plants, especially very small conifers like arborvitae that you've just planted, take a look on those needles, look for these little bags. You might be able to kind of smack them off the branches onto some paper to sample them. But just be aware that the little ones are out. They're a little bit more susceptible to pesticides. Once they get older, they're going to be very difficult to control, and they're going to be very voracious. So this is the time to scout if you're worried about bagworms. Also, the fall webworms, even though it's spring, once it starts to warm up, and by the middle of the summer, you're going to really be seeing them. These actually are going to be starting to make the small webs in trees, and you're going to find these little tiny caterpillars starting out. So be on the lookout for those. If you want to remove them from the plants, you can usually then just take the small colony out and destroy it just with pruning and whatnot. But the bagworms, the webworms, lots of other caterpillars are starting to wake up or hatch and grow up to feed on plants. And oftentimes we see them start seeing the bigger ones in the middle of the summer, but they're obviously don't get born that way. They're going to be small at first. In addition to caterpillars, some sawflies are going to be more prevalent now. These are ones on elms. But some of the other plants they'll feed on are sometimes pines, but also hibiscus and roses. So you may be seeing some sawflies out there as well. And don't confuse them with caterpillars. They have a single eye lens, and they have 6 or more pairs of these fleshy prolegs on their abdomen, compared to 5 or fewer in caterpillars. Of course, one of the banes of most gardeners are the Japanese beetles. This is the time of year when they become active. They're beginning to emerge and going to plants. They feed on flower petals, but also the leaves. The adults do of lots of different plants, but mostly I see them on roses and grapes and things like that. So be on the lookout for them. If there's only a few on a plant, they could be hand removed. They don't bite, they don't hurt. They could just be hand removed and drowned or something like that, frozen, however you want to kill them, I guess. but they will be around for about a month or so, maybe a couple months, and then they're going to be kind of dying out as they've already mated and laid their eggs in the ground, which will then become larvae, the white grubs that will grow up through the fall, overwinter, and then begin feeding again in the spring to become new adults next year. Of course, there's lots of bugs that are going to be out and about, true bugs, lace bugs, and flea hoppers, and lots of little plant bugs. In fact, I just saw on Twitter, somebody showed some pictures of mint from up north where there are 4-line plant bug damage on it. So these are out and about and feeding on various plants. So depending on the type of plant you have, you may have a certain suite of these pests. The carpenter bees have been very active, of course, already, but you may be seeing the tiger bee flies. These are very large black flies that look very menacing, but are harmless, completely harmless to humans. They are not so harmless to carpenter bees. They are actually a specialist, a parasitoid on carpenter bees. They lay their eggs in the tunnels, and their larvae then attach on the larvae of the carpenter bees, feed on them, and then they emerge as these very large flies. So this is the time you might be seeing those. Of course, outside the garden and attacking you are going to be mosquitoes and ticks. I was out the other day taking photos, and I was attacked by lots and lots of mosquitoes. Definitely wear repellents. DEET is very good for mosquito and biting fly control. It's not as good for tick or not control, repellency, but it's not as good as for tick repellency. But for ticks, impregnating your shoes, outdoor shoes that you use for gardening or being outside, and pants with permethrin is a good way to keep ticks and chiggers from crawling up and biting you. They will, once they come in contact with clothing that's impregnated with that or treated with that, they will die. And of course, the problem with this year, time of year, or what I like to say, the great thing about this time of year is everything is out. So there's lots and lots of activity from all different insects. And other arthropods. That's why I can't cover everything, of course. So you may be seeing weird things like this fungus feeding moth or this lizard beetle or all these other critters out there. Again, as always, if you find something you're not sure about, you can contact me, send photos through our system, and we can help you identify whether it's a friend, foe, or just someone hanging around a little bit. And finally, I just want to make sure that everybody is aware. You might be getting calls about giant hornets or invasive hornets or things like that. You won't see any northern giant hornets, aka the murder hornets, here in North Carolina. Right now, what people are mostly seeing are going to be European hornets. They are almost as large as the other giant hornets, very, very impressive insects, but are different in several ways. So be sure you know how to tell the difference between these two groups. The main differences are in the giant hornets, the northern giant hornets. I should change that because that's not an official common name. They have smaller eyes in relation to the head, and their head is entirely yellow with very clean stripes on the abdomen. And in European hornets, the back of the head is this kind of brownish red, as is much of the body. And they have these stripes on their abdomen that have these little teardrops or spots associated with them. So if anybody is worried that they have non-native aggressive northern giant hornets, they are almost certainly going to be European hornets now, or maybe some other large hornet-looking insect, but we do not have the northern giant hornets here at the moment. And I will look at some questions while I turn it over to Mike.
So Matt, the Asian giant hornet is now northern giant hornet.
Yes, that is the
Okay.
that's the now the approved common name. It's called northern giant
Okay.
hornet. Or you could call it Vesper mandarinia, which is the scientific name. That's the most accurate thing. And I'm not sure about the program up in the Pacific Northwest where it's been found. But as far as I know, they really haven't been finding it anymore. So it may be at some point eradicated, and then maybe people will stop talking about it. But we'll see.
Okay.
But anyway, we've got lots of comparative websites comparing all these different things, and maybe I'll throw a link in there while Mike is talking.
Sounds good. Yeah, I made a note to make sure I get those in the resources after the webinar because you've got some great comparison charts for us. All right, Mike, turning it to you.
All right, Matt, though there was I guess it was a comment rather than a question about the use of basil for attracting or maintaining a wasp parasite or parasitoid or predator of Japanese beetle larvae. Did you see that?
Yes, yeah, I'm seeing a few other questions, and maybe I'll just address them very quickly. Well, I'll address them in the chat while you get going, but I'll respond to the chat.
All right, such a good boss. He doesn't want to cut into my time. All right, share a screen here. I don't know if I have that new version of seeing what we're sharing, but I think I finally, after all these years, mastered getting the right screen to share. And this looks like it, disease bolos for late June, which I'll just abbreviate to June in the rest of the slides. Talking about first of all, I wanted to start with a cautionary tale. This was not a June sample. This came from, I think it was August, quite a few years ago now, but it was at a golf course landscape bed, and they had a monthly rotation of fungicide drenches. They had 1 month it was one set of fungicide, another month it was another. So back and forth between two fungicides or two groups of fungicides. And in spite of that, they had something like 40% of their impatiens showing a problem. And it turns out this in the sample, and it turns out that what was really going on was root knot nematodes. Of course, no fungicide is going to help against that. So sometimes you're battling against one thing preventively, and it's something else that sneaks up and gets you. And I present this because I had kind of nematodes on my mind with a sample that came in recently and delved into that a little bit. So I want to have our first bolos be about nematodes. And I also am going to toss up a poll here. Let's see, A through E. So see what folks think about this particular question. According to the North Carolina Department of Agriculture and Consumer Services, how many kinds of plant parasitic nematodes are reported in North Carolina? Is it 4, 14, 34, 54, or 94? And just give folks a quick moment. The picture there is actually a photo that I took, of a foliar nematode. So most of our plant feeding nematodes are in the roots, of course, but there are a couple that do cause problems in aerial plant parts, including the aphelincoides, a few species of aphelincoides that are known as foliar nematodes. So it looks like folks have kind of already tossed their opinions in here. We'll end the poll and share the results. And it's kind of a bell curve there, excuse me, as far as what people thought, but the most people thinking it was either 34 to 54, but actually it turns out that the 8 of you who answered 94 were correct. That is the number of plant parasitic nematode species or genera when there's not a species associated. Most of those were listed as species, as I recall, that are in that list. Now, of that, I think it's about 12 fall into the category that I'm going to talk about real quick, which are the root knot nematodes, the ones that cause galls on the roots. And those tend to be our more important, more damaging ones in North Carolina. But of those 12 that are reported, there's 4 really that are our main, well, now 5 with Meletigoin enterolobica, but enterolobii, but that is not one we've seen in home gardens yet. That's been a field crop problem so far. So our main 4 is still going to be the southern, the peanut, the Javanese, and the northern root knot nematode. And they can have different host preferences, as a matter of fact, which makes it a little bit trickier to deal with them. These sort of luminescent kind of pear-shaped objects are the females of root knot nematode that I dissected out of a root, and you can see some of the eggs also hanging around there, because the females inside the roots just become basically egg-laying factories in their adult life, it's the egg sacs that are the overwintering stage, by the way. Among the plants that you want to be on the lookout for root knot nematode problems in the woody area, boxwoods, we see quite often with root knot nematode, as well as the lesion nematodes and the spiral nematodes. Gardenia can have very serious root knot problems, And one of the few diseases that we see on our figs, or edible figs in the landscape, is the root knot nematode. In fact, we just got an image sample this week of a fig tree that had classic nematode galls. Among the flowers, if you want to call them flowers, liriope and also mondo grass would be good candidates to be on the lookout for that. And begonia and impatiens. Now, I want to draw your attention to the two photographs here. On impatiens, on begonia, you're going to see pretty large galls, but on the liriope, you may have to look carefully. In fact, they're usually smaller than the normal storage root swellings on that plant. So if you look here, right at the tip of that finger, there is one of those nematode galls on that liriope root. They're not always that tiny, but they are definitely something smaller than you would see on some other hosts. In the vegetable garden, well, sweet potato, that's mainly because of the field, but in the vegetable garden, it would be the tomatoes and the cucurbits would be our main concern with root knot, but they also occur on beans, on beets, spinach, okra, so they have quite a wide host range. Now, since we're just doing bolos and saying what to be on the lookout for, we're not going to really talk about control measures, but I'll drop into the chat box real quick here a couple of publications that you can use to reference for management of root knot nematodes. Let's see here. So there's one from, or the set of notes from the North Carolina Department of Agriculture and Consumer Services. They call them Nemanotes, and number 12 is illustrated there, and then a very thorough publication from the University of Florida. So you'll find both of those links in the chat. Moving on with other bolos in the landscape and garden for the coming month. This is actually a photograph that was just submitted yesterday. I think it was yesterday, of dogwood. And if you look carefully, you can see several things going on. There's kind of a bronzing to the leaf. I've usually seen it as more of kind of a grayish coloration that happens, also some distortion. But if you look carefully, especially where the leaves are a little bit of an angle to the sun, you can see that this in fact is powdery mildew that's going on here. So that's one of the ones where you have to sometimes examine carefully to be sure that you've got powdery mildew when you're dealing with dogwood. On other hosts, though, it's quite common. So while you're out picking the Japanese beetles off of your crepe myrtles, if you have a sensitive, a susceptible cultivar, then you're probably going to also be seeing pretty obviously some powdery mildew there, and of course on quite a number of other hosts in the landscape. We will be soon seeing the quince rust Esia on our calorie pears. So those papery coated structures inside which the spores are produced, dry orange spores. And they can be so heavy, you'll notice the coating here on the leaves. And I've even seen a photograph of where you get an orange cast to the pavement below because there's so many spores being released. These don't really infect the leaves, but you get it mostly on the fruit, but you can also get some stem infections with a characteristic swelling and then the Esia being produced as well there. And that will persist. So even when the other evidence is gone later in the year, you may see some of the swollen stems. I've seen this, for example, on Amelanchier, which gets a gymnosporangium. I'm not sure which particular species, but service berry is another thing that you'll be seeing this phenomenon happening on soon. And for the maples, this is going to be a good time of year to be seeing some of the different leaf spots that develop, such as the Philisticta leaf spot, and also the group that we call anthracnoses. So the fungus Discula, Aureobasidium, these kind of things will be showing up on our Japanese and other maples in the landscape. Turning to the vegetable garden, so a reminder from last month when we talked about problems in cucurbits, and one of them was downy mildew, which is not a true fungus, a fungus-like organism. Starts out on cucumber anyway, bright yellow angular spots that then would turn brown. And on the right there, that's an example on squash. You'll notice how they later turn a brown color because the spots actually die. You can also see some powdery mildew mixed in there with the downy mildew in that photograph. With tomatoes, the first fruits will be coming on, and often those first clusters will have blossom end rot from a localized calcium deficiency. Not uncommon to see that on the early fruit and then not on the later, but not an infectious disease. We do, though, have a number of infectious diseases that are important on our tomatoes in the garden. Septoria leaf spot on the left there. It can get much, much worse than what's shown there, often affecting a large number of the lower leaves. You could actually pick off those leaves up to about 1/3 of the foliage of the plant if you want to try and kind of reduce the inoculum there. Whereas with tomato spotted wilt virus, that's systemic in the plant, and the spotted leaves will tend to be up in the new growth. Nothing you can do about picking that off. And also, as we start to see fruit set, we may see some of the blotches and ring spot symptoms coming from the tomato spotted wilt virus. And then the bane of our tomatoes is, of course, the southern bacterial wilts caused by Ralstonia solanacearum, which will cause the loss, of course, in short order of the entire plant, a soil-borne bacterium. Switching over to the fruits, in apples, we mentioned, I think last month, that the pycnial stage could be confused for insects laid on the upper surface of the leaf is going to be in the, those are going to be now passé. We've already had that stage, and we'll start to see on the undersides of those same spots, the esia developing, which will have this fringe here, peridium it's called, and then inside the orangish spores, those esia spores that are going to disperse and, if they're lucky, land on a juniper somewhere to complete the life cycle. The spots, the symptoms are pretty characteristic when you see this on apple, on crabapple, of bright yellow to orange spots on the leaves. And it seems to me, just casually, that we have quite a bit of fire blight out there this year. We've had a couple of samples come into the clinic, and I've seen it 1 or 2 places as well, just out and about. This particular sample from Granville County this month, and when it came in, it had the very typical shepherd's crook of the symptom of the fire blight, and that tested positive for the bacterium and Rhinia amylovera. You've got to make sure in the active season like this that you prune way below that. So go a whole foot into what appears to be clean wood to make sure you get ahead of that bacterium. And as a general bolo, just in the landscape beds, we may start seeing our dog vomit slime mold, Fuligo septica. Starts out, as I've showed before on the program, as a frothy yellow material. It develops then to kind of a pinkish dull crust that, if broken open, will release the spores that can blow off and then continue the life cycle. This is completely harmless to both us, pets, and plants, but it does sometimes cause alarm when it shows up in our landscape beds. And finally, I'll just give one bolo for the turf, which is brown patch. Now that the warm weather is going to set in, it's been so nice, but we will start seeing those warmer temperatures, and that tends to stress out the cool season grasses like our fescues, and Rhizoctonia solani takes full advantage. This irregular sort of spotting here caused by the brown patch fungus. I even heard that you can smell this on a moist morning. I can't say that I've ever been able to do that, but just a little interesting tidbit there. And if you want any more bolos, just go to our website, click on the bolo link, scroll down to June, and feast. With that, I'll take a quick look over at the chat. Let's see. All this good insect stuff here that Matt was. Are root knot nematodes seen in the Piedmont? Aren't they mainly in sandy soils? In fact, they are seen across the state. So we see them a lot in Wake County. We've seen quite a few in Guilford County. I didn't check to see what host. Certainly, probably the number one maybe would be down there in New Hanover County. They'll cause more stress on the sandy soils because just it's impeding root functions. You've got less water holding capability, less nutrient holding capability in those soils, but they can be found across the state. Let's see. All right, unless I'm missing something, I don't see any more questions.
Okay, thank you, Mike. Excellent. Thank you, Matt, for answering things along the way. I will.
Can I ask a question?
Certainly.
Okay, I planted mustard seed trying to get rid of my root knot nematodes. What do I do with it? Do I pull it out? Do I let it sit there? Whatever.
That needs to be incorporated into the soil. So the chemicals that are produced by our, brassica, the full advantage of that, my understanding, is you want to incorporate that into the soil. There may still be some information about that on the Department of Ag website in one of their NEMA notes, although they tend to emphasize more the marigold, the use of the marigolds. And those, of course, have to be planted as a full crop across the garden plot and not just sprinkled in among the vegetables in order for that to be effective. But yeah, when you're using the mustard family for root knot control, the way I've heard it is that gets incorporated into the soil.
So then is it safe to plant something then in the soil?
I wouldn't see why not. Of course, any kind of fresh plant material, you've got to give it some time to decompose a little bit, or it may tie up some of the nitrogen, I suppose. I don't have direct experience with that exact system of doing that with the mustard, so I can't say for sure. Maybe someone else out there has that piece of information.
Thank you.
Yeah,
I think it's going to help suppress the populations. I wouldn't recommend planting really susceptible and sensitive plants in those beds, even after you've used the mustard. It's just going to help you long-term suppress those populations. But I'd still look at if it's vegetables, like some of the resistant varieties of tomatoes and things like that. And that's also going to help suppress and increase in those root knot nematode populations.
And right, you're never going to completely get rid of them. And you have to use the multi-pronged approach of crop rotation, resistant varieties, some of these suppressive plants that you can put on and so forth.
Is there a handy dandy list of these resistant vegetables and things?
Check that link out from the Florida publication. I think they do have some of those. I don't know how up to date they are. And there's also either there or linked from, no, there are on there some tables of, let's say in the flower bed garden, just a long list of different plants and whether they're susceptible or resistant or immune. So you can use that in your decision making if you've got them in the flower bed.
Thank you.
All right. Thank you, Mike. And we will continue on with our resource spotlight, which is our feature every month where I try to tell you a little bit about a resource you might know about or might be new to us. So let's see. This month's resource spotlight was actually inspired by an email that I received last week. And this is the email. It was an NC State pest alert. And it was highlighting a new article that had come out actually about armyworms and Japanese beetles. And when I got this email, it made me wonder, do our master gardeners know that they could also receive these emails? And do they know how to subscribe to them? So this month, I want to share with you how you can get these as well if you're interested. So when I clicked on the article about Japanese beetles, it took me to this page. This is the NC State Extension turf file page. And this is where the article was posted. You can see right there in the news and updates section. And because I'm subscribed to get these updates, anytime there's a new news or update in this turf file page, I will get an email. And when I clicked on the article, it opened up the article. This was written by Dr. Bilson, and it was sharing the fact that fall armyworms and Japanese beetles, their activity has actually been reported in Lee and Wake counties, and the activity is a little bit earlier than last year. So it just let us know that we should be looking out and scouting a little bit earlier for Japanese beetles and fall armyworms. Now, if this type of article is of interest to you and you'd like to be notified by email, it's down here in the right-hand corner that you can enter your email address and hit submit. And you are now subscribed to set up those email, to receive those email updates when new content is posted. However, that's just from the turf file page. There is a whole lot more to be aware of. So today's resource spotlight is really this link that's highlighted in yellow, this subscribe link, which takes you to the subscription page for NC State weekly extension updates. And here's a picture of the page that you land on, and it gives you the opportunity to choose what updates you might like to receive. Now, they're called weekly extension updates, and this is a whole list of the subject areas that you can choose from. I selected a few as an example, just to show you. Some of the ones that I subscribe to are entomology, integrated pest management, turf files, and one I wanted to call your attention to is this one, new publications and fact sheets. This is going to send you an email anytime across NC State Extension. There's some new publications that have been released or fact sheets. And you can scan this list and see what's of interest. Definitely some of them don't apply. There may be a new fact sheet, something relating to poultry, but there may also be a new fact sheet related to Japanese beetles. So it's just another way to stay up to date on things. So you simply check the boxes of which ones you're interested in. You enter your email address down here in the right-hand corner. You hit subscribe, and you are subscribed to all of those at once. Now, I did want to call your attention that it does say weekly extension updates, but it's really not it's not really weekly. In my experience, it's more like once a month, once every 6 weeks or so. It's just when a new article has been posted on one of those sites. So it should not overwhelm your email. And, you know, if anytime you start subscribing, it's not what you're interested in, when the email comes through, here's the email again, you can just unsubscribe down here at the bottom, and you've been taken off that list for that particular page. So anyway, I hope that you will check out that link and choose the areas that you're interested in and, you know, get those notices. All right, let's move on to announcements for the month. The first one is to remind you of a continuing education opportunity coming up really soon. On June 8th, we're going to have Basil Camus, who is from a local tree care company here in Raleigh called Leaf and Limb. And he's going to share with us 8 ways that gardeners can help heal the earth. So he's going to talk to us about incorporating building healthy soils and proper pruning techniques for trees, keeping our trees healthy, things like trading turf grass for more of a prairie environment. So I hope that you can join us, and you can sign up right on the internet calendar. Okay, we have kicked off our new Learn, Grow, Share Showcase series. And these webinars will feature projects presented by you, by Master Gardener volunteers and extension agents. And this is our chance to learn about inspiring projects that are going around the state and to connect with one another. So we would love to hear from you about what you would like to learn about. And also, we would love to hear if you have a project that you'd like to share with others. So this is a link to a quick survey to let us know, hey, what are you interested in? And also if you've got a project that you're willing to share. So we invite you to fill that survey out. We also hope you saw our new quarterly statewide e-newsletter. We're also calling this Learn, Grow, Share. The first issue came out on April 28th. And everyone who has an EMG intranet account has been automatically subscribed to receive this. So if you are active on the EMG intranet, you should have gotten this newsletter, and it would have come from this address, ncstate-emg@lists.ncsu. So if you didn't receive it, maybe check your spam. But if you did not receive it, you can go to this link down the bottom here. Is going to take you to the page on the intranet that has all of our past issues. So you can read the April 28th issue, and it will also let you send us a updated email, or basically it's a request to get added to the list, and I'll make sure you're added to that. Now, there's another way to also stay up to date on things that are happening with the Master Gardener program across the state, and that is our NCEMGV Google group. So this is more for breaking news. So we send out updates and announcements as they happen. So this is if you want to receive a little bit more frequent updates from us. This is an optional Google group that you may join. So you must subscribe to receive these, and the messages will come from this address. And so if you'd like to subscribe, make sure that your email address is up to date. Here's a link for you to join the Google group, and you'll get those updates. Okay, last month I mentioned that our own Matt Bertone was featured in Our State magazine. And I also mentioned that the Master Gardener Volunteers of Durham County, led by Deborah Pilkington, was featured for their work on container gardens. And so I wanted you to have the links to those articles. They're here, but also the entire issue of that magazine featured articles on extension. And so I've put the link to all of those articles here, and it's a great issue. It really expands your knowledge of the breadth of extension. There's an article on how extension helped respond after Hurricane Fred. There's an article on pole beans and how pole beans is a crop that's coming back, sort of experiencing a renaissance. There's an article on Mark Wethington, who is the director of the J.C. Ralston Arboretum, and it talks about his travels, his plant explorations. So hope that you can enjoy that article, or that whole issue, really. Okay, now I just wanted to take a moment to recognize you all, because the Extension Master Gardener program is 50 years old this year. This is a 5-minute video that was made by Washington State University in celebration of the 50th anniversary of the Master Gardener program. And the Master Gardener program did start in Washington State in 1973. But here in North Carolina, the program began in 1979. The first garden hotline was in New Hanover County, and the first training class was held in Wake County. And in just a few years after it started, so we started in 1979, by 1982, there were already 110 volunteers in 5 counties. And just to look at where we are today, now we have over 3,500 volunteers in 83 counties across North Carolina. So it's a tradition that we can be proud of, and I hope that you'll enjoy this short video. Okay, there is a Lunch and Learn webinar coming up next week regarding common pests and diseases of food crops. And so this might be interesting for you to attend. It's being hosted by the Center for Environmental Farming Systems, which is a partnership between NC State, NCA&T, and the North Carolina Department of Agriculture and Consumer Services. And CEPS develops and promotes just and equitable food and farming systems. So Melissa Bell and John Little will be presenting some common pests of North Carolina vegetable crops and some options for control. There is a new citizen science opportunity that we'd love to have you participate in. This has been called to our attention thanks to Amanda Wilkins, who is the horticulture agent in Lee County. She's working on this project. The Great Southeast Pollinator Census is going to take place across Georgia, South Carolina, and North Carolina. And this is a one-time contribution program where they're inviting citizen scientists of all ages to observe pollinators during a discrete 2-day sampling period. And the sample period will be August 18th and 19th. So to learn more about this, there's going to be an informational webinar on June 7th. And you would follow this link to sign up to attend that webinar. But also keep in mind, I know that that webinar is going to be recorded. So if you can't make it on June 7th, you could still watch the recording and get involved in August. All right, that brings us to the finale and a look ahead at next month. So next month, Mike and Matt and our team, we're going to focus on updating you on invasive plants, pests, and pathogens. So we're all going to focus on some of the pathogens that pests and pathogens and plants that we've spoken of in the past as being invasive. Where are they now? What is an update for you as the growing season continues? So we look forward to seeing you in June for that program. And we thank you all for being here today. I think it was a great amount of information. Thank Colleen Doherty again and Mike and Matt for their preparation as always. And we wish you a really good day. I will collect all the links as I do, and you will be able to access the recording from today and all the resources with the live links we posted on the EMG Intranet Plants, Pests, and Pathogens page. And it's usually up by tomorrow. So everything will be there for you. And great to see you all. Thank you.